Welcome to another video of the Paper 1-2 series. This is a 9700 February-March 2018 Paper 1-2. If you are new to this channel, you know what to do. Like, comment, subscribe, and finally watch and learn. Without wasting any more time, let's get into it. Question 1. The eyepiece lens of a microscope can be fitted with an eyepiece graticule. Which of these statements about eyepiece graticules are correct? 1. They measure the actual length of cells in micrometers. 2. They help biologists to draw cells with correct proportions. 3. They change in size when the objective lens is changed from 10 to 40. Eyepiece graticule remains fixed in size no matter the magnification. Therefore, statement 3 is incorrect. Statement 1. Measuring the actual length involves calibration. You cannot measure the actual length simply by looking at the eyepiece graticule. Eyepiece graticule only helps when drawing plan diagrams or drawing cells with correct proportions. Therefore, the answer is D, 2 only. Question 2. A student was asked to use a scale bar shown to calculate the magnification of a cell on a photomicrograph. Which method could the student use to calculate the magnification of the cell? Given is the actual size, which is 2 micrometers. And how do you calculate magnification? Magnification is nothing but image size upon the actual size. Let's go to the options A, B, C and D. A. Divide the diameter of the cell by the length of the scale bar, both measured in the same units of length. That is incorrect, because in this method they are basically telling us to divide the actual size by the image size. However, the magnification formula is image size upon actual size, not the other way around. Therefore, method A is incorrect. Method B. Measure the diameter of the cell in millimeters, multiply by 2000. This method is obviously incorrect because multiplying by 2000 is not involved in this formula, therefore is incorrect. C. Measure the length of the scale bar in millimeters, convert to micrometers and divide by 2. Yes, that is correct, because once you measure the length of the scale bar in millimeters, you get the image size. However, the image size is in millimeters and the given actual size is in micrometers. In order to substitute the values into the given formula, you need to make it into a single same unit. Therefore, you convert the obtained image size into micrometers, and then when you divide by 2, which is basically the actual size, you get the magnification. Therefore, C is the correct choice. Let's look at D. Measure the length of the scale bar in millimeters, convert to micrometers and multiply. That is incorrect, because magnification is image size upon actual size, not image size into actual size. Question 3. Which eyepiece and objective lens combination of light microscope allows the greatest number of cells in a field of view to be seen? Greatest number of cells in a field of view can be seen only when the magnification is the least, because lower the magnification, more the area that can be seen. More the area, that means more cells are visible. As the more you magnify, the area becomes less, or basically you're just viewing a smaller area but more magnified. However, the least magnified it is, the more field of view can be seen, and hence the more number of cells that can be seen. Therefore, the combination should give the least magnification, which is 5 into 10, which is 50. Therefore, A gives us the least magnification, hence will allow the greatest number of cells. Question 4. Which row correctly matches each cell structure with its function? Microtubules and rough endoplasmic reticulum. Let's look at option A. Synthesizes amino acids under rough endoplasmic reticulum. That is incorrect, because rough endoplasmic reticulum is mainly concerned in the production of protein and transport of proteins. It has nothing to do with synthesizing amino acids. B. Rough endoplasmic reticulum produces ribosomes. That is also incorrect. Ribosome production occurs in the nucleus, specifically in the nucleolus. Therefore, B is incorrect. Option C. Rough endoplasmic reticulum transports proteins. Yes, that is correct. Microtubules form the spindle during prophase. That is also correct. Therefore, C does show the correct matching of the cell structure with its function. Why is option D incorrect? Because there is a mention of rough endoplasmic reticulum making triglycerides and phospholipids, which is not at all correct. Question 5. Which animal cells would have the most extensive Golgi bodies? Over here, you simply have to look out for the keyword goblet cells, because goblet cells produce and secrete mucus, and they have an extensive number of endoplasmic reticulum as well as Golgi bodies. It is because they are needed to synthesize the proteins found in mucus. Hence, the best possible answer is goblet cells. If the answer consists of goblet cells, that will have the most extensive Golgi bodies. Question 6. Which of these processes will require ATP? 1. Transport of water in the xylem. 2. Semi-conservative replication of DNA. 3. Facilitated diffusion of amino acids into the cell. Facilitated diffusion doesn't require ATP at all. Therefore, answers having 3 in it are obviously incorrect, leaving us between options B and D. Let's look at process 2. Semi-conservative replication. Yes, it does require ATP because ATP is required in bringing the activated nucleotides, if you recall the process of DNA replication. And 1. Transport of water in the xylem. Well, transport of water is mainly concerned with cohesion and adhesion forces. It doesn't need ATP. Therefore, the best possible answer is 2 only, that is D. Question 7. Which concentrations could be produced by a serial dilution of an 8% glucose solution? The starting glucose solution concentration is 8%, and serial dilution is where the dilution factor is constant throughout. Let's look at A. From 8% to 4%, yes, it is halved. Then 4% to 2%, again it is halved. 2% to 1, 1 to 0.5, and 0.5 to 0.25%. Yes, it does follow a constant dilution factor throughout. Therefore, A should be produced by a serial dilution. Hence, A is the correct answer. Why are the others incorrect? Let's look at B. 4 to 3, 3 to 2, 2 to 1, and 1 to 0. Yes, it is constant. However, the initial is 8. And from 8 to 4, there is a decrease of 4%. But from 4% onwards, there is a decrease of 1%. Therefore, it is not a constant dilution factor. And similarly, C and D also do not have a constant dilution factor if you compare the differences between the following concentrations. Question 8. A student wrote these statements about polysaccharides. 1. Amylose is formed from condensation reactions between beta-glucose monomers. 2. Branches in amylopectin molecules form between carbon atoms 1 and 4 on alpha-glucose molecules. 3. In unbranched beta-glucose chains, each monomer is rotated 180 degrees relative to its adjacent monomer. Which of these statements are correct? Key thing you must know is that amylose and amylopectin are both polymers of alpha-glucose only, and amylose is not a branch structure whereas amylopectin is. And how are those branches formed? Branches are formed due to the presence of 1,6 linkages. Therefore, if you go to the statements 1, 2 and 3, statement 1 is obviously incorrect. Why? Because it mentions beta-glucose monomers. However, amylose is formed from alpha-glucose. Therefore, 1 is incorrect, leaving us with the answer being either C or D. Let's look at statement 2. Branches are formed between carbon atoms 1 and 4 on alpha-glucose molecules. That is incorrect because branches are formed between carbon atoms 1 and 6, not 1 and 4. Therefore, statement 2 is also incorrect, leaving us with the best possible answer being D, 3 only. And yes, in unbranched beta-glucose chains, each monomer is rotated 180 degrees relative to its adjacent monomer. If you look at cellulose, cellulose does show this key property. Question 9. Which diagram correctly shows the formation of a peptide bond between two amino acids? Formation of a peptide bond occurs between a carboxyl group and an H2 group. Hence, diagram B is obviously incorrect, considering between diagrams A, C and D. D is also incorrect because there is a presence of C double bond O single bond NH2, which is not present in amino acids. Therefore, D is incorrect. Let's look at A and C. OH is removed from COOH group that is bound to a carbon atom followed by a NH2 group, not any other group. Therefore, A should be the correct diagram. Why is C incorrect? Because C shows the removal of an OH group from a carbon atom that is bound to two carbon atoms and then a NH2 group. However, A shows only one carbon atom and then an NH2 group. Therefore, A is the correct choice. Again, OH is removed from a carboxyl group that is bound to one carbon atom followed by NH2. However, C shows the removal of an OH group from a carboxyl group bound to two carbon atoms followed by NH2. Therefore, the correct diagram is A. Question 10. Sugars with a ring structure can also have a linear structure. Which sugar molecules could be represented by the linear structure shown in the diagram? The given structure consists of 6 carbon atoms. If you count the carbon atoms in this given diagram, it has 6 carbon atoms. Hence, it cannot be ribose or deoxyribose. As ribose and deoxyribose, they are pentose sugars consisting of only 5 carbon atoms. Hence, answers A, C and D are incorrect, leaving us with the best possible answer being B, alpha glucose only. And yes, alpha glucose is a hexose sugar consisting of 6 carbon atoms. Question 11. Which molecules are globular proteins? 1. Amylase, 2. Hemoglobin, 3. DNA polymerase. A key thing you must remember is that all enzymes, no matter what the enzyme is, are globular proteins. And both amylase and DNA polymerase are enzymes, as well as hemoglobin is also a globular protein due to its soluble nature in water and arrangement of hydrophilic and hydrophobic groups. Hydrophilic groups are arranged towards the outside region and hydrophobic groups towards the center, and that makes it a globular protein, giving it a spherical shape. Therefore, the answer is A, 1, 2 and 3. Question 12. When hydrolyzed, which molecules have products containing a carboxyl group? 1. Phospholipids, 2. Polysaccharides, 3. Proteins. Polysaccharides do not have a carboxyl group or a COOH group, whereas others do, as phospholipids consist of fatty acid tails and proteins have amino acid chains. All acids have a carboxyl group. Remember this key point, all acids have a carboxyl group. Therefore, since phospholipids consist of fatty acid tails and proteins are made up of amino acids, they do contain a carboxyl group as they are all acids. Hence, the answer is B, 1 and 3. Polysaccharides do not have any acid chains in it, therefore do not have a carboxyl group. Question 13. Lysosomes contain many different hydrolytic enzymes that may act within cells, intracellular enzymes, or outside cells, extracellular enzymes. Which process must occur in order for lysosomal enzymes to act outside the cell? To act outside the cell and not inside, it needs to be packaged into vesicles and transported to the cell surface membrane, causing its release, which is known as exocytosis. Therefore, the process that must occur in order for lysosomal enzymes to act outside the cell should be exocytosis, because exocytosis is where vesicles are involved and the releasing of substances out of a cell. And since the enzymes need to be released from the cell to act outside the cell, it needs to be exocytosis. Question 14. The diagram shows an enzyme, its substrate, and an enzyme-substrate complex. Which statement shows how the substrate is able to enter the active site of the enzyme? Question refers to how the substrate is able to enter the active site. And as you can see, initially before the substrate enters the active site of the enzyme, the shape of the active site and the shape of the substrate are not complementary in nature due to different sizes. They do not have the same size and are not complementary in shape. Therefore, if you go to the answers A, B, C and D, answer B is incorrect because B mentions the shape of the active site and the shape of the substrate being complementary. However, it is not. In order for the substrate to bind with the enzyme, it should be that the shape of the active site changes in order to bind with the substrate. Then only the substrate is able to enter the active site. Otherwise, since it is not complementary, it cannot bind. In order to bind with the substrate, the enzyme should cause a change in the enzyme's shape or a change in the active site. Therefore, the best possible answer is A. Contact between the substrate and the enzyme causes a change in the enzyme's shape. The other answers do not describe how the substrate is able to enter the active site. Question 15. What could be used to calculate the rate of an enzyme catalyzed reaction? 1. The appearance of product. 2. The disappearance of substrate. 3. The Michaelis-Menten constant. The rate of a reaction is generally governed by the amount of substance or substrate used up or the amount of product formed, and both over a period of time. Therefore, what could be used to calculate the rate of an enzyme catalyzed reaction should be 1 and 2 only. 1 being the appearance of product, 2 being the disappearance of substrate. 3, that is the Michaelis-Menten constant, has nothing to do with the rate of an enzyme catalyzed reaction because Michaelis-Menten constant is concerned with enzyme affinities towards a specific substrate. It doesn't govern the rate of an enzyme catalyzed reaction. Hence, the answer is A, 1 and 2 only. Question 16. Cholesterol is an integral component of the cell surface membrane. Which statement about cholesterol is correct? A key thing you must know is that cholesterol, it regulates FSF, F being the fluidity, S being the stability, another F being flexibility. And cholesterol is a molecule that is hydrophobic in nature. It doesn't allow any polar substances to pass through the membrane. And if you go to the answers, A, it allows ions to pass freely through the cell surface membrane is incorrect because ions are polar in nature, and cholesterol, as I mentioned, doesn't allow any polar substances to pass through. B, it has a hydrophobic head, that is correct, and a hydrophilic tail, that is incorrect because cholesterol molecules do not have hydrophilic paths. C, it helps to regulate the fluidity, that is correct because cholesterol, it regulates fluidity, stability as well as flexibility. Hence the answer is C. And if you go to D, it reduces the mechanical stability, that is incorrect because it increases the mechanical stability. Question 17. Which type of bond in phospholipids has a role in increasing the fluidity of cell surface membranes? Increasing the fluidity of cell surface membranes is concerned with either cholesterol molecules or double bonds in fatty acid tails of phospholipids. Therefore, since this question refers to bonds, the answer should be any answer that has a double bond, and that double bond should be between two carbon atoms only. Therefore, the best possible answer being B, carbon-carbon double bond. D shows a double bond between carbon and an oxygen atom, which is incorrect. The double bond should be between two carbon atoms. Hence, the answer is B. Question 18. Which of these substances can pass directly through cell surface membranes without using a carrier protein or a channel protein? That means substances that can diffuse directly across the cell surface membrane without using any transport proteins. Let's look at 1, 2 and 3. 1. Calcium 2 plus ion. This is an ion, and ions are polar in nature or they are charged. Therefore, they will not be able to cross directly to the cell surface membrane as polar substances cannot pass through the hydrophobic fatty acid tails of the cell surface membrane. Let's go to 2. Carbon dioxide. Yes, carbon dioxide is a non-polar molecule and it is a small molecule. Therefore, it is able to diffuse directly to the cell surface membrane without using any transport proteins. 3. 3 represents glucose, and glucose is a large molecule, and large molecules are not allowed to cross directly through the membrane. It has to use some form of transport protein. Therefore, it does not pass directly through the membrane. And since the question refers to what substances pass directly through the membrane, it should be 2 only, hence leaving the answer as D. Question 19. Companion cells use ATP to move hydrogen ions out of the cell and co-transporter proteins to allow hydrogen ions to return with sucrose molecules. Which two processes are involved in this movement? By reading this question, you should be able to understand that this is regarding the loading of sucrose, which, if you recall the chapter of transport in plants, loading of sucrose does involve active transport, hence the answer being either A or B. And the question itself mentions return of sucrose involves co-transporter proteins, and if it involves transport proteins, it is facilitated diffusion, not simple diffusion, because simple diffusion doesn't involve any transport proteins, whereas facilitated diffusion does. Therefore, the answer is B, active transport and facilitated diffusion. Question 20. How many copies of each different DNA molecule will be found in a cell at the start of each of these stages of the mitotic cell cycle? G2 of interphase. Now G2 phase is after S phase, which is where DNA replication occurs. DNA replication occurs in the S phase of interphase, and since G2 is after S phase, it will definitely have 2 copies, because once DNA replicates, 2 copies are produced. Therefore, the answer being either C or D. And the question refers to the start of each of these stages, not end. Hence, for prophase and cytokinesis, it will also be 2. Cytokinesis is where cell splits, and at the end it would be 1 copy. However, since the question refers to the start of cytokinesis, it will be 2 copies. Hence, the answer is D. 2 for G2 of interphase, 2 for prophase, and 2 for cytokinesis. Question 21. 3 parts of a chromosome and their functions are listed. The parts being centromere, histone proteins, and telomere. The functions being packages DNA into compact structures, holds 2 chromatids together, prevents loss of genes. Which combination is correct? Centromere definitely holds the two chromatids together. If you go to the paths and the functions, P1 matches to F2, therefore that is one possible combination. However, P1 and F2 is not given as a combination in the answers. Therefore, let's go to another combination. Telomere, it prevents the loss of genes, therefore P3 matches to F3. And yes, P3 and F3 is given as a combination as an answer, therefore the answer is D, P3 and F3. The other combinations are incorrect, because the correct combinations are centromere holds two chromatids together, histone proteins packages DNA into compact structures, telomere prevents loss of genes, F1 matching to P2, F2 matching to P1, F3 matching to P3. Hence the answer is D, P3 and F3. Question 22. The graph shows various distance measurements taken from metaphase of mitosis onwards. The graphs are to scale when compared to one another. Keyword over here is taken from metaphase of mitosis onwards. That means after metaphase. And what follows metaphase? Anaphase, telophase, and finally cytokinesis. Therefore, let's look at the graphs x, y, and z. In x, the distance for a period of time seems to be constant. Afterwards, the distance seems to decrease. For y, the distance is constant throughout, and for z, the distance increases. Let's look at the question. Which row correctly identifies the distance measurement for each graph? Obviously, y should represent the distance between the two poles at the opposite ends of the cell, because that distance doesn't change at all. Therefore, y is the distance between opposite ends of the pole, leaving us with the only possible answer being C. Distance between poles of spindle being y, x being the distance of centromeres from poles of spindle, and z being the distance between sister chromatids. Let's look at the graphs again. For z, after metaphase, anaphase occurs, which is chromatid separation. Hence, the distance between the sister chromatids starts to increase, and that is represented by z. Let's look at x. The distance of centromeres from poles of spindle decreases. Hence, it should be x. Therefore, C is the correct choice. Distance of centromeres from poles of spindle represents x. Distance between poles of spindle represents y. Distance between sister chromatids represents z. Question 23. A culture of bacteria was allowed to reproduce using nucleotides containing the heavy isotope of nitrogen. After several generations, all of the bacterial DNA molecules contain heavy nitrogen. DNA was extracted from a sample of the culture, mixed with cesium chloride solution, and spun at high speeds in a centrifuge. In this process, DNA molecules of different masses separate into bands at different positions in the centrifuge tube. The heavier the DNA molecules, the closer to the bottom of the centrifuge tube that a band forms. The diagram shows the position of the DNA molecules containing heavy nitrogen in the centrifuge tube. The culture of bacteria was then allowed to reproduce using nucleotides containing the light isotope of nitrogen, 14N. DNA samples were taken and separated by centrifugation after the bacteria had divided once and again after the bacteria had divided twice. In which positions would the DNA be found after the cells had divided once and after the cells had divided twice? This, although a long question, you basically have to break it down into key parts. First, they would place it into 15N, which is the heavy nitrogen isotope. And they would make sure they obtain several generations of the bacterial DNA molecules. And all of those bacterial DNA molecules would contain heavy nitrogen only. After that, when they spun it into a centrifuge, you get what is represented by M in the given diagram. That is of 15N only. Then they placed it into a culture of bacteria containing 14 nitrogen only and allowed it to reproduce. Now, after it reproduces once, the new generation of bacteria will contain both 14N and 15N. Basically, 100% of hybrid DNA, that is 14N and 15N. That is one molecule consisting of one strand from 14N and one strand from 15N. That is a hybrid DNA molecule. That is for the first division, and after it had divided twice, there will be half at L and half at K. Why is that? Because some would be hybrid DNA and some would be only 14N. Because it is placed in a culture containing light nitrogen only. So after dividing once, all should be at L, because the first generation is of hybrid DNA and all would consist of hybrid DNA, that is one strand of 14N and one strand of 15N. After the first division, when placed into a culture containing the light nitrogen, that is 14N. Hence the answer is only C, because only C talks about all at L after dividing once, and after dividing twice, half at K and half at L. Therefore, the best possible answer being C. Question 24. The diagram represents a molecule of ATP. What are the components of ATP labeled P, Q and R? P consists of adenine sugar, which is a purine. Purines are guanine and adenine. Q consists of a pentose sugar, which is ribose. And R is basically 3 phosphate groups, because ATP stands for adenosine triphosphate, and by the meaning of tri, it means 3 phosphate groups. And if you go to the answers, R represents phosphates, not a phosphate group and not phosphorus. Therefore, the answer being either A or D. And Q is not deoxyribose but pentose, because ATP, remember it carefully, ATP has a pentose sugar which is only of ribose, not deoxyribose, thereby leaving us with the best possible answer being D. P being purine, Q being pentose, R being phosphates. Question 25. The table shows 3 anticodons for different amino acids. Which DNA triplet on the DNA template strand codes for the amino acid serine? A key thing you should remember is that the bases on the DNA molecule are the same as that of anticodon, except that U in anticodon is replaced by T in DNA. And since they are requesting for the amino acid serine, which is the anticodon UCA, the DNA triplet would be U being replaced by T, hence starting with T, C being as it is, and A being as it is. Therefore, the DNA triplet is TCA. Hence, the answer is B. Question 26. The photomicrograph shows a transport tissue in the stem of a grapevine. What is the structure labeled T? T represents an end wall or a sieve plate. And if you go to the answers, A. Companion cell, B. Sieve plate, C. Sieve tube element, D. Xylem vessel element. The question refers to the structure labeled T, and T is a label of an end wall or a sieve plate. Hence, the best possible answer being B, sieve plate. Why not sieve tube element? Because sieve plate is a structure that is present in the sieve tube element, and the structure labeled T is pointing towards the sieve plate, not towards the vessel. Hence, the answer is B. Question 27. The diagram shows transverse sections of parts of a plant. Transport tissues are labeled 1 to 6, which row shows tissues that mainly transport water and tissues that mainly transport sucrose. Tissues that mainly transport water are the xylem, and tissues that transport sucrose are the phloem. And I have labeled towards the sides which one is which. 1, 4 and 6 being xylem, 2, 3 and 5 being phloem. You must remember where the xylem and phloem are placed in different transverse sections of parts of a plant, of a leaf, stem and root. And then this question becomes nothing but easy. Mainly transport water being, out of the given options, 4 and 6. Mainly transport sucrose being 2 and 3. Because A, B and C consists of incorrect combinations. D is the possible combination that would give you the correct answer. Question 28. What will increase the rate of transpiration? 1. Increase in the humidity. 2. Increase in the light intensity. 3. Decrease in the temperature. 4. Decrease in the wind speed. Rate of transpiration increases due to increase in wind speed, increase in light intensity, increase in temperature, and finally decrease in humidity. Hence, increase in the humidity won't increase the rate of transpiration. Therefore, 1 is incorrect, leaving us with the answer being either C or D. Let's look at C. 2 only, that is, increase in the light intensity. Yes, that is correct. Increase in light intensity does increase the rate of transpiration. Let's look at D. 4 only, decrease in the wind speed. That is incorrect, because increase in wind speed will lead to increase in rate of transpiration, not decrease in the wind speed. Therefore, the best possible answer being C, 2 only. Question 29. Which properties of water are important for transport in xylem? 1. Cohesion. 2. Adhesion. 3. High energy requirement for evaporation. 4. Good solvent properties. In xylem, water transport occurs, which requires cohesion and adhesion forces. Therefore, the answer must contain both cohesion and adhesion, leaving us with the best possible answer being only A, because only A has both 1 and 2. The other answers do not have both 1 and 2. Therefore, A is the correct choice. 1, 2 and 4. Cohesion, adhesion and good solvent properties. Why good solvent properties? Because xylem transports water and mineral ions. And in order to transport mineral ions must be able to be soluble in water. And water should have good solvent properties. Therefore, A is the answer. Question 30. Which statement about the blood flow in cardiac cycle is correct? A. Blood flows into the aorta through the semilunar valve due to contraction of the right ventricle. That is incorrect. It flows into the aorta from the left ventricle, not right. If you recall the structure of the heart and how the cardiac cycle occurs, blood will flow into the aorta from the left ventricle. Blood will flow into the pulmonary artery from the right ventricle. Let's look at B. Blood flows in the left atrium through the pulmonary artery where the wall of the left atrium relaxes. Blood flows into left atrium through the pulmonary vein, not the artery. Therefore, B is incorrect. C. Blood flows into the right atrium through the vena cava when the wall of the right atrium relaxes. That is correct. Why is D incorrect? Blood flows into the right ventricle through the semilunar valve due to contraction of the right atrium. Blood flows through the atrioventricular valve, not semilunar valve. The semilunar valves are only present between the ventricles and either the aorta or the pulmonary artery, and it is the atrioventricular valve that separates the atrium and ventricle, not the semilunar valve. Therefore, D is incorrect, leaving us with the best possible answer being C. Question 31. Which of these statements about the formation of hemoglobinic acid are correct? 1. It depends on the formation of carbaminohemoglobin. 2. It removes excess hydrogen ions, preventing the blood becoming too acidic. 3. It is linked to the action of carbonic anhydrase. 1 is obviously incorrect because it doesn't depend on the formation of carbaminohemoglobin at all. It depends when hydrogen ions binds to red blood cells, leading to hemoglobinic acid, and it doesn't depend necessarily on carbaminohemoglobin. Therefore, answers having 1 in it are incorrect, leaving us with the best possible answer being D, 2 and 3 only. It removes excess hydrogen ions, preventing the blood becoming too acidic. Yes. 3. It is linked to the action of carbonic anhydrase. That is also correct, because hydrogen ions is formed after carbonic anhydrase carries out its reaction. Therefore, D is the answer. Question 32. What can combine with the heme group of a hemoglobin molecule? 1. Oxygen. 2. Carbon dioxide. 3. Carbon monoxide. In this kind of question, you must remember that only oxygen and carbon monoxide combines with the heme group of a hemoglobin molecule. Carbon dioxide doesn't combine with the heme group. Therefore, answers having 2 in it are incorrect, and answers should consist of 1 and 3. Hence, the answer is B, 1 and 3 only, because yes, both oxygen and carbon monoxide can bind with the heme group of a hemoglobin molecule. Question 33. Which factors would help a person to adjust from living at a low altitude to living at a high altitude? 1. Formation of fewer red blood cells. 2. An increase in the oxygen carrying capacity of the blood. 3. An increase in the output of blood by the heart. Living at high altitude causes an increase in the red blood cell count, not decrease in red blood cells. Since factor 1 talks about formation of fewer red blood cells, that is incorrect, because actually living at high altitude leads to formation of more red blood cells. Therefore, answers A, B and C are incorrect because all 3 have factor 1 in it. Therefore, leaving us with the answer being D, 2 and 3 only. An increase in the oxygen carrying capacity, yes. An increase in the output of blood by the heart, that is also correct. Question 34. Chronic obstructive pulmonary disease includes bronchitis and emphysema. Which symptom is specific to emphysema? The keyword over here is specific to emphysema only, not to bronchitis as well. A. Excess mucus secretion by the goblet cells. B. Inflammation of the bronchial epithelium. C. Loss of elasticity of the alveolar walls. D. Thickening of the smooth muscle of the bronchi. Emphysema is concerned with elastin in the walls being broken down and hence loss of elasticity. Therefore, the symptom that is specific to emphysema is C. Loss of elasticity of the alveolar walls. The other symptoms are not specific to emphysema but can also include bronchitis as well. Therefore, the answer is C. Loss of elasticity of the alveolar walls. Question 35. What identifies the structures present in bronchus? All trachea, bronchus and bronchioles contain cilia. Only trachea and bronchus have cartilage, as well as both trachea and bronchus consist of goblet cells. The one that doesn't have goblet cells are the bronchioles. Since the question refers to bronchus, it will have all 3: cartilage in the form of irregular blocks or plates of cartilage, cilia is also present, and goblet cells, yes, they are present. Therefore, the answer is C, because all 3 are present in a bronchus. Question 36. Which of these terms can be used to describe the role of mosquitoes in the transmission of malaria? 1. Malarial parasite. 2. Pathogen. 3. Vector. The role of the mosquitoes is just to act as a vector only. Therefore, terms 1 and 2 are incorrect because the question refers to the role of mosquitoes. And the role of mosquitoes is to act as a vector, leaving us with the answer being D, 3 only. Question 37. Bacteria that infect skin wounds can become resistant to the antibiotic commonly used to kill them. Which statement explains how these bacteria could become resistant to the antibiotic? Antibiotic resistance is mainly linked with mutations. And if you go to the answers A, B, C and D, A. Exposure to the antibiotic causes a change in the bacterial cell wall, preventing the antibiotic from working. Purpose of antibiotic is to target the bacterial cell wall. Hence, it should cause a change in the bacterial cell wall. Therefore, it doesn't prevent the antibiotic from working. Hence, A is incorrect. B. Other bacteria that normally protect the skin against infection are killed by the antibiotic, allowing the infectious bacteria to survive. This statement doesn't involve mutations anywhere in the given statement. Therefore, it is incorrect because antibiotic resistance is mainly concerned with its own mutations. C. Random mutations in DNA allow some of the bacteria to survive in the presence of the antibiotic. Yes, that is a possible statement because mutations can occur anytime, anywhere based on certain factors such as exposure to X-rays or any ionizing radiation, and mutations in its own DNA could lead to forming resistant strains. Therefore, C is a possible statement. D. The antibiotic causes the bacterial DNA to mutate, allowing the bacteria to survive in the presence of the antibiotic. Antibiotics do not cause bacteria to mutate at all, hence D is incorrect, leaving us with the answer being C. Question 38. Some children are born with severe combined immunodeficiency. Children with this inherited disease do not normally have any T lymphocytes and suffer from many infectious diseases. How may these children be cured from SCID? A. Transfusion of antibodies. Antibodies do not carry out the role of T lymphocytes but only the role of B lymphocytes. Therefore, A is incorrect as these children do not have T lymphocytes. B. Treatment with stem cells. Yes, stem cell therapy can help produce T lymphocytes and therefore aid in the role of T lymphocytes acting on pathogens and preventing the formation of infectious diseases. Therefore, B is a possible answer. C. Use of different antibiotics. Antibiotics only work on bacteria and not all infectious diseases because infectious diseases can be caused by viruses too. Hence, C is incorrect. D. Vaccination against all diseases doesn't help as no T lymphocytes are produced that can respond. Therefore, B is the possible answer. Question 39. The hybridoma method is used for the production of monoclonal antibodies. Which two types of cell are used in this method? A. Stem cell and B lymphocyte. B. Stem cell and T lymphocyte. C. Tumor cell and B lymphocyte. D. Tumor cell and T lymphocyte. In the hybridoma method, tumor or cancer cell and B lymphocytes are involved. The tumor cell is involved because it provides the ability to divide continuously, whereas the B lymphocyte is involved to produce antibodies that cannot divide multiple times. And once you combine these two cells, you would get hybridoma cells that are able to both produce antibodies as well as divide continuously. Hence, the answer is C. Tumor cell and B lymphocyte. Question 40. A vaccine is used to create artificial active immunity. After being given a vaccine, it will take a period of time before a person develops long-term immunity against a disease. Which statement about this period of time explains this delay? A. No memory cells have been produced from B lymphocytes. B. No plasma cells have been produced from B lymphocytes. C. The primary immune response has not produced enough antibodies. D. The secondary immune response has not produced enough antibodies. When given the vaccine, it takes some time to develop memory cells that form part of the immunological memory. Hence, the delay is mainly due to no memory cells being present, because the body should carry out its own immune response in order to fight the pathogen that is injected through the form of vaccination. It could be a live organism or it could be a dead strain of pathogen. Therefore, since no memory cells are produced, the body will take some time to fight the pathogen injected into the body through the form of vaccination, and that causes the delay because no memory cells are produced. Hence, the answer is A. No memory cells have been produced from B lymphocytes. That is why once memory cells are produced, when the secondary immune response occurs, it occurs quickly and antibody productions occur at a higher rate, hence leading to very less delay. However, over here, since it refers to a person being given a vaccine, it will take some time, and that time is taken in order to produce memory cells. And the answer should consist of no memory cells being present in the body, that is, no memory cells have been produced from B lymphocytes. And that is the end of this paper. Hope you found this video useful. If you did, hit that like button, comment and subscribe if you haven't already. Do let me know down in the comment section below which paper I should be doing next. Thank you for your time and stay tuned for more.